This is the BioCentury Show. Brought to you by BioEquity Europe. BioEquity is Europe's premier international showcase for financial dealmakers and biopharma executives to meet rising biotechs.
Hello, welcome to the BioCentury Show. I am Simone Fishburn, Editor-in-Chief of BioCentury. Delighted to be joined today by Merdad Parsey, the Chief Medical Officer of Gilead. Merdad joined Gilead in 2019 after serving as SVP of Early Clinical Development at Genentech. And prior to that was CEO of 3V Biosciences, now Sagimet, and had several development roles in different various biotechs. So we're going to start out by talking about some of Gilead's own programs. And then I also want to have some discussion about critical issues in drug development more broadly. And I think you'll be able in that, Merdad, to draw on some of your experiences as well in working in smaller companies, because you've really sat on different tables or different sides of the table. I think, Merdad, welcome again. Let's start with oncology, because I think those are the biggest questions and interest people have. Gilead made its name in infectious disease with HIV. That still dominates its sales. But much of the talk is about oncology, and your CAR-T product, Yescarta, from the Kite acquisition, just passed $1 billion in sales, and it leads the pack. So it is now a blockbuster. So very simple question. Are you now an oncology company? And how far are you in that transition?
Hi, Simone. Thanks for the invitation. I'm really excited to be here. So yeah, well, look, I think we are both, right? We're both a virology company and an oncology company, and hopefully an inflammation company as well. As time goes by, our strategy had always been that we wanted to build on the strength of Gilead, that we didn't want to substitute one for the other, but we wanted to build on that strength. And in many ways, it was actually part of the draw for people to come to Gilead, was coming to a place that had such a great established track record, and the background that we have. It allowed people to have sort of the sense of stability and the sense of that we were building something meaningful for the long run. And certainly the Kite acquisition was a great starting point for that. As we started to build oncology. Now that predated my time and predated Dan's time, but it was sort of a great foundation upon which to really build our oncology programs. It's certainly been a challenge over the past few years, sort of starting from there, and made doubly so by being during the pandemic. You know, I started in November 2019, and December is when those first cases in China started showing up. And of course, it was a great time for our virology strength to be put to the test with remdesivir as we went through that, really in the early part of 2020. And during that same time, we started building our oncology portfolio and the team. And it really took both. I'd say right now we're maybe about halfway, a little bit more in terms of our overall build out. My hope is that we continue to grow to be a sustainable company across the board. We really feel the need to invest in our early pipeline as well as the later pipeline, so that we're a sustainable company for the long run.
So let's dig into that a little bit. Now, so when I look at your pipeline, I see, and your deal activity, right? I see a tremendous amount of investment and interest in CAR-Ts. Mm-hmm. I see a very anchor product, perhaps, in Trodelvy, your ADC. I see a program in TIGIT, which we can talk about, and then another one in CD49. The TIGIT one, of course, comes from the Arcus deal. The CD47 one comes from the 47 deal. How do you think about building out in those areas? You don't seem to be heavily into bispecifics. Is that an area you'll go into? Are you going to do more ADCs? Tell me how you're thinking about sort of building out that portfolio.
Sure. I think it's a number of things. The first, I think to look at it, we were very strategic in our early acquisitions because we felt that in order to really establish ourselves as an oncology company, we had to build some late products. We had to bring in some late products. And that was really behind.
And is that still an emphasis for you to bring in more late stage products?
Not really. I think certainly from an opportunistic standpoint, we're always going to look there. But initially, the strategy was very much that we needed to build an anchor point. And I think you're absolutely right that Trodelvy really formed the basis of our anchor point. And we now are building around that. And we wanted to make sure that we got some those assets. And you mentioned magrolimab, our CD47, and domvanalimab, our TIGIT antibody, which again, I would argue are somewhat de-risked farther along, clearly still a ways to go, but really can help form the underpinning and a real basis for the long-term pipeline. And now we really feel the need to fill the early pipeline. We've done some of that. You've seen us do a number of deals there. That is obviously a much more challenging space where things don't always work out, as you know, right? A lot of promising things, but they don't always work out. And so we really want to build that part of our portfolio to really complement as we launch, obviously Trodelvy's already launched, but as we launch some of the other programs, that we have more things coming along to help complement. Now, it's not, to your point, we're not taking a haphazard approach. We're really trying to focus in oncology in a few areas of biology. We're really focused on IO, the immuno-oncology space, for example, TIGIT. We're looking at intrinsic cell death. That's sort of the Trodelvy pillar. And then there's a pillar around the tumor microenvironment where we're also trying to focus, and you've seen us do a few deals there in the early biology. I think that's proven to be more challenging for everyone, the tumor microenvironment. But our goal is to really try to fill each of those verticals, if you will, with an idea that by combining those modalities, we will be able to really make a big difference. We're approaching it less specifically around a particular type of modality.
Right. So it seems that you're building it based on scientific sort of, I don't know if I would say mechanisms, but sort of schools, as it were.
Yeah, more biologically driven than modality driven. And
right.
and we want to leverage all the innovation that's happening on the modality side. You mentioned ADCs. We love Trodelvy, and it's been really fun to watch the ADC space really get exciting again and really build out. So we do hope that there'll be more opportunities there, but we're not going to be an ADC company, if that makes sense, right? We're going to use it as it makes sense in the right place.
Okay, so let's come back to CAR Ts in a minute, because there's a special set of skill sets that go with that. But presumably, you know, there will be people listening who might be interested in partnering with Gilead. And, you know, what I'm hearing is you're going to go after, let's say, the science, but what in terms of building up your own capabilities? You've now got capabilities based on an ADC. How transferable is the in-house knowledge versus relying on a partner if you want to bring in a bispecific or a nucleic acid kind of therapy or whatever? So how do you feel about that balance?
Yeah, I think those are great examples of things that we are interested in and are doing. We've done some partnerships on the bispecific side, and that's certainly some place where we see it going. You know, and for us, I think it's really around this idea around not just engaging T cells, which I think is a really interesting place, and we're definitely active there. But we see the future as sort of looking at modalities where you are able to target, for example, an antibody to a specific cell type and pursue a specific biology, because I think that's sort of where the science is going. One of the areas where we think that we will have new therapeutics that will allow us to get to better therapeutic indices, right? To get better efficacy with less toxicity over time. In a sense, that's what the ADC world is. But I would argue that if you look at things like T-cell engagers or other types of protein Fab fragments, those sorts of technologies, or even when you look in the small molecule space, where you're looking at protein degraders or different sorts of small molecule technologies that you can look at protein-protein interactions. All of those things, I think, ultimately are designed to get to not only new targets, but also to better therapeutic index. So you can really be much more targeted in your therapeutic and hopefully bring fewer side effects. But that's sort of where we see things headed.
Right. I mean, that's how I see most of the new modalities. They segregate into whether they give you access to a new target, right, or whether they reduce your, give you a better therapeutic index, let's say therapeutic window for your target. Let's stay with science in a minute. And I want to go back to something you talked about before. When you referred to TIGIT and even CD47, you used the word de-risked. So let me pick up on that a little bit, because TIGIT's been a very tricky target. And, not just for Gilead, I'm talking about across drug development, right? Yeah. And, outside of LAG-3, we really haven't seen in the post-PD-1 era new checkpoint targets. And, LAG-3 is proved out, if you want to say that that's de-risked now. So we're still really waiting for them to deliver. So what is the principal challenge? You think, is it finding the right way to interrogate these targets? We haven't quite got the right either patient population or trial design, or do you think that there's less there? Is there less fruit than people hoped?
It's such a great question. It's something we've all been struggling with for quite some time. I wish I had a magic answer. I do think, look how long it took us to get to the first checkpoint inhibitor, right? We started with IL-2 many years ago in prostate cancer, right? And it took us 20 years. I can't do the math, but a long time to get to PD-1 and PD-L1 inhibitors. So, those sorts of major breakthroughs don't happen quickly. And you're right. I think that we have a unique challenge with immuno-oncology agents now. I think in part because we're looking for them to bring activity on top of a highly active therapy, right? So you're really looking to add to PD-1, PD-L1 activity. That's pretty challenging. Those are really effective molecules. And what I think you see a lot of the field struggling with is in early development. So often you have to go to randomized studies. You have to go to randomized patient populations, because differentiating the monotherapy results of a PD-1, PD-L1 from a combination with a TIGIT or a LAG-3 or any other immuno-oncology agent is challenging. And so it takes a little bit more effort. Having said that, I think TIGIT is a good example of one where both given the Roche Genentech data and the data we've shown together with Arcus, has evidence of clinical activity. And we've seen that the outcomes are improved when you add it to a PD-1 or a PD-L1. Merck has shown the same thing. There is a lot of data now that support the role of TIGIT. What I think where we are today in terms of TIGIT is how broad, how deep will that TIGIT additivity be? Will we be adding to people who are already PD-1, PD-L1 responsive and getting to better outcomes there? Will we be broadening the remit, if you will, or the depth of response, say to people who are say PD-L1 1 to 49? Can we get deeper responses in those patients? Are we going to turn somebody who had what we call a cold tumor or PD-L1 PD-1 poorly responsive tumor into a responsive tumor? That's a bit more challenging. I think that's where the jury's still out. But I think for me, I believe it's de-risked because I think the clinical data so far has been pretty, pretty clear that TIGIT adds something to the PD-1 activity.
All right, in a minute, we're going to go to a break, but I want to ask you just one last thing on this topic and go back to CAR-Ts. Now, obviously, I think Gilead, has pretty much proved itself. You seem to have manufacturing figured out at least as well as your competitors from where we sit at BioCentury. And as I talked about, you've got a fair amount of deal activity. A lot of that is on early stage stuff. So at the same time, Christy Shaw, the CEO of the Kite unit, recently announced her departure. And a lot of the innovation there seems to be quite early stage. Solid tumors, allogeneics, those are still really challenging. So what do you see as next for that unit? And what do you think is the next step change along with CAR-Ts that patients should expect?
Yeah, so look, I think Christy did an amazing job along with the rest of the Kite team and really helping shepherd CAR-Ts from a place where there was unreliable delivery, challenging data, to a place where I think it's firmly established now. You look at the data in DLBCL and the 5-year outcomes, and I think that's been incredibly powerful to make that transition. And the Kite team does an incredible job of making sure that patients who get apheresed are going to get treated. The reliability of getting the cells to patients has been really great. And that neither of those is trivial. You know, we take it for granted that you can get a small molecule or you can get an antibody fairly easily, but there's no shortage of challenges there in terms of cell therapy. So I think it was really necessary to establish that firm beachhead, if you will, really to solidify the reliability and the data for cell therapy in a way that now we can build from there. There are a lot of challenges still left. I agree with you that moving into solid tumors or into allogeneic has been challenging and probably will be for a while. And one variable, which is sort of the ability to make the cells, I think the Kite team has really, in a sense, taken off the table. We can now think about what is the right biology, what's the right approach to get to these things. I think it remains to be seen. And so our approach there has been to go in a measured way, right? Making sure that we solidify establishing Yescarta and Tecartus's place, making sure that anyone who needs those therapies is able to get it in a reliable way, while looking for generating the data internally or externally. And you've seen us do some partnerships, you've seen us do some acquisitions, but we're looking to generate the data. I mean, we really need to see the data develop to really expand the remit. Whether you're talking about into different tumor types, we talked about solids, but even myeloma, or even into new therapeutic areas. As you know, the work on CAR-Ts being applicable potentially in inflammation is ongoing. And so I do think there's really quite a future for cell-based therapy writ large. And we need to figure out, both from a tolerability standpoint as well as an applicability standpoint, where that balance lies, and we're determined to be at the forefront of that. I think that's an important commitment, an area for us.
All right, we're going to take a brief break, and when we come back, we'll talk a little bit about Gilead in virology and some more broader drug development questions. Thank you.
Thanks.
The 23rd Bioequity Europe Conference heads to Dublin, Ireland in May 2023 with a program focused on how to create a new playbook for biotech success in Europe. In 2022, over 800 industry leaders from 30 countries joined Bioequity in Milan, Italy for 2+ days of strategic panels, one-to-one partnering meetings, and C-level networking. More than 150 biotechs selected by BioCentury presented their story to investors and potential partners. Don't wait, 2 of the last 3 in-person Bioequity conferences sold out. Register today and join BioCentury EBD in our Dublin Regional Host Committee at the industry's premier C-suite and investor conference. Visit bioequityeurope.com for more information.
All right, back with Merdad Parsey from Gilead at the BioCentury show. Merdad, I want to just spend a couple of minutes on virology. I think it's not really a question, the impact that Gilead has made on HIV and even on HCV as well, actually. But I do want to ask you, we talked a lot about oncology. I want to ask you about the growth path in virology And I think for many people, let's talk a little bit, because you're still working in HIV. I think a lot of people sort of think about it and they're like, well, all right, they've turned a death sentence into a chronic disease. And what is the big unmet need now? So tell us what you're working on and where you see this as important.
Sure. I mean, my perspective, it's interesting because you mentioned turning a death sentence into a chronic illness. My perspective, when I remember being in medical school when HIV was first coming out, and being a house officer when, if you were admitted with AIDS at the time, it was really a death sentence. And to see how much it's changed, it's been an unbelievable transition in my professional career. To see that and to be part of that now, to be able to be part of that is really fantastic for me. Look, I think the sad reality is that there are still too many people getting infected with HIV, and there are too many people who have a hard time with their medication regimens, and I think we should be at a place. The WHO and we have really been trying to get to eradicating HIV, right? This is a fully attainable goal. And it's very frustrating to me as a clinician that we haven't been able to get there. So for us, what's in our control is what can we do? Well, for one thing, I think we feel that there are ways where we can make it easier for people living with HIV to be treated, whether you're somebody who's highly treatment experienced, who may have multiple drugs that you're resistant to. I think bringing out lenacapavir, a first-in-class capsid inhibitor, really presents a huge leap forward, where you can now have a new class that hopefully can help treat those people. There's another aspect, which is this long-acting part, where there is an important group of people who don't want to be reminded each day that they are infected, and the ability to potentially treat them once a week, once a month, once every 3 months, once every 6 months, or even out to a year, is really important to that population of people, where both from a compliance and a resistance and a psychological standpoint, all those things, having long-acting treatments is going to be important. So we think there's a lot of opportunity there to bring really great options for people living with HIV. And then very importantly, on the prevention side, I think that if you think about how we're going to get to eradication, it really is not only treating the people who are living with HIV, but preventing HIV. And right now, only about a quarter of people who should be getting prevention are actually getting it. And we think that by bringing out ways of preventing HIV globally that are easy to take, have great compliance, and again, lenacapavir we think is going to fill a great role there, is really going to be part and parcel of being able to get to a point where we're not talking about increasing HIV rates, even in the US. I mean, that to me is really an incredibly frustrating place to be as a modern society, that we're still talking about this.
Yeah, well, actually, I could go in so many directions from this. How long are we going to be talking about the death rates from COVID and so on? But I won't go there. We'll save that for another day, Merdad, because I want to, you previously were in a virology company. Clearly, this is something that is close to you, but I want to tap a little bit into your experience having been first CEO and in other situations in smaller companies. Now, several of the initiatives from FDA have come up recently that are really good for patients, but really difficult for small companies. I'm talking about things like requiring dose optimization trials early in cancer, or requiring them at all instead of maximum tolerated dose. Having clinical trials represent the patient population demographically, obviously an important thing for patients and for trials. But all of these things make it much harder for small companies to do trials. And I know that Gilead likes to partner with small companies as well. So that's obviously one outlet. But how do you think, having been in that, in the driver's seat in such a place, how do you think about these challenges and what it means for their ability to really drive programs?
Well, I think, first of all, it's really important to remember that getting to those goals like diversity in trials is really important for us. If we think about it as a community, as a society, making sure that we're getting understanding how well drugs work in different populations and making sure that we're delivering those benefits to whether it's people with breast cancer, TNBC, something we're really focused on, or HIV, all of those things across the board are critically important. So I do think they're incredibly valuable for patients to be able to do that. And yes, look, the way I think about it is as we innovate, as new treatments come out, the bar continues to get higher. That's a good thing. We are at a place where treating cancer has gotten, we're not done clearly, but it's gotten so much better over the past 20, 30, 40 years. The bar is higher. The bar is higher for us in terms of what we need to do and in terms of what benefit we need to bring to people. So I actually think it's completely appropriate for us to be raising the bar as we go along and have options for patients. So from a small company standpoint, I think there are probably a couple of lessons in there. The first is you really have to bring something that you think is going to move the needle. It's really going to bring an important benefit to patients. That is probably the biggest driver and the biggest challenge for all of us. The days of sort of, incremental benefits, I think, are numbered. And so I think that's a big part of it. And then secondly, I do think it does put some pressure to partner a little bit earlier. I don't think those pressures that you mentioned, diversity and dose optimization, are critical in very, very early development. I would say up to proof of concept, you probably don't need to do as much of that. Ideally, you would, but in your phase 1b studies, for example, if we're focusing on oncology. But I do think that as you get into now the later stages of development, as you start to move to phase 2 and phase 3, the burden does go up appropriately for developing there. And I think we just have to accept that that's going to take a more intensive effort, and often that'll mean earlier partnership. And from my perspective, we're happy to be part of that ecosystem to enable that, recognizing that it's got a challenge. It's going to be more difficult to get things across the finish line.
Well, you've given me a great segue to my penultimate question. You talked about raising the bar. So, I don't think it's hard to, few people could really argue that the pace of innovation in the last couple of decades has just been remarkable. And, the things that have gone, I don't think many people could have predicted we'd be where we are with biology. Conversely, the pace of drug development, not to mention cost, has not gotten much better. And how do you see this? Do you see this changing? Is there a reason that we might see it changing? And basically, I'm really asking, are we going to get better and faster at making drugs?
Boy, is that a 2-minute answer that you want? Well, let me start by saying I don't know. And part of the reason I say that is, look, I think that inflammation, in a sense, provides an interesting case study for that, okay, where we started with TNF inhibitors coming out. Not started, but that's where a real step change in the treatment of inflammatory diseases happened, right? And that was back in the 90s and early 2000s, and it really made a big impact. Now people's standard of care is to be on a TNF inhibitor in so many diseases, and it's really become, you don't even think about it. And what the consequence of that is it's actually raised the bar for the benefit you need to bring to people with rheumatoid arthritis or inflammatory bowel disease, where you really need to show benefit on top of an active agent. And it parallels for me, the PD-1 story in many ways, where the bar goes up. It's put more pressure on us to think about how we're going to innovate in that space and how we're going to bring benefit. Combinations have become part of the answer there. I think we believe in others that you're going to have to show benefit on top of those sorts of standard of care, just like we were talking about earlier with PD-1 inhibitors in oncology. So that's harder. I mean, let's just accept that it's going to be harder. There are places where we haven't made a lot of headway. There are diseases where the amount of innovation, the amount of benefit that we've brought has not been as great. And I think those are places where you can probably move faster. You're not adding on to an already effective drug. So I do think the pace will change. But what I'm reassured by is just in my career, I guess I'm an old guy now, so it doesn't really, I can't say that anymore. But I've seen a couple of really big step changes, right? TNF inhibitors, PD-1 inhibitors, where we really thought we knew the answers before they hit, right? And then when they hit, it really had a step change. So to me, I think I'm by nature a pessimist, but empirically, we've seen so many step changes in treatment that have happened over the past just 20, 30 years that I believe with the pace of technology and modalities and things like that, that I am optimistic that, you have a step change and then an incremental improvement and then another step change and incremental improvement. And that's how science works. And I'm excited to see. I don't know what the next one's going to be. Cell therapy has been one for a lot of patients with DLBCL. I don't know what the next one's going to be, but I'm confident there is another one coming, and that's what I get excited about coming to work every day.
Well, I said penultimate, so I do get to ask you one quick other question. Staying with your optimism, what are you particularly excited about, either on the scientific or clinical front? What might we be talking differently about? Let's end on the optimistic.
Yeah. Look, I think the pace of innovation and technology has been remarkable, right? We are able to do single cell proteomics. We're able to look in microenvironments at cell-cell interactions. So I think there are breakthroughs in basic biology that are happening now that were not possible 10 years ago even. And similarly, I think on the modality side, we're able to go after, there was so long that we had undruggable targets. And now with the way we are able to come up with modalities, whether looking at small molecules for protein-protein interactions or mRNAs, 3 years ago, no one thought mRNAs were going to be really effective. Here we are, we're vaccinating everybody with mRNAs. I think those new modalities are going to bring options for interrogating that biology in a way that we didn't have. And so those are I think it's the intersection of those two things, really the novel biology, and then our ability to actually go and either interrupt or augment the biology in a way that we didn't have access to, that I think is really going to be where the innovation is going to come from over the next, decade or so.
Clearly, we could talk for a long while, but we're going to have to do it again instead. Merdad, thank you very much for a great conversation, and we certainly look forward to the next time.
Absolutely, my pleasure. Thanks for having me, Simone.
The BioCentury Show is brought to you by BioEquity Europe. BioEquity is Europe's premier international showcase for financial dealmakers and biopharma executives to meet rising biotechs.